Hello students, this is topic 3 lecture on cell culture and medium. In this lecture, I will be focusing on in vivo versus in vitro culture. At the end of this lecture, you should be able to know the differences between in vivo animal cells and in vitro cell culture, the advantages and disadvantages of in vitro cell culture, and also the major types of in vitro cell culture. Section 3.11 Differences between in vivo animal cell and in vitro cell culture. In vivo in the Latin language means within the living. So it basically involves studying cells within their native organism. And this method actually provides the most accurate representation of how cells actually behave in their physiological context. On the other hand, in vitro is a Latin word for in glass, meaning it involves growing living cells in a very highly controlled, non-living environment. Okay, so you're basically growing and maintaining cells outside of their natural environment, typically in a laboratory setting. So this kind of cell culture is often carried out by growing these cells in either a 2D dimensional plane, such as on glass or plastic petri dishes, or even the cell culture flask that you all have had your hands on during the practical sessions. In vivo systems refer to that biological systems or processes that actually occur within living organisms, such as animals or humans. So these systems are inherently three-dimensional because living organisms exist and they also operate in a three-dimensional space. And within these 3D systems, cells are actually organized and oriented in very specific ways within the organs and tissues in order for them to function efficiently. So this organization is really crucial for the proper functioning of that particular organism. And you can see that in vivo, different cell types within an organ would then have very specialized functions, and that their specific arrangements actually allow them to carry out these functions effectively. They can communicate within each other to coordinate their activities, and the way they are arranged actually also facilitate this communication through various kind of molecular signaling pathways. So for example, like organs such as liver or kidneys, they have specific arrangement of their cells that actually ensure that they actually get efficient blood supply, and also exchange of nutrients and waste products are really taking place efficiently. However, if you were to compare it with an in vitro cell culture, it's really very different because in vitro cell culture focuses specifically on the growth and study of individual cells or small groups of cells. In vitro cell culture involves cells being grown as a monolayer, and these monolayer systems, what actually happens is that the cells are plated onto a flat surface, such as a petri dish or a tissue culture flask, where they actually adhere and then they proliferate. And then as they divide, these cells will then spread out and they will form a single layer, with each cell will be having direct contact with the underlying substrate. This monolayer of cell culture actually provides a very simplified model, but they are actually very valuable model system because it allows us to study basic cellular processes and also conduct experiments in a very controlled environment outside that complexity of living organisms. When a cell line is actually formed, okay, it typically originates from a single cell or a very small population of cells that you actually isolate and culture in vitro. And over time, these cells will then divide and proliferate, and then they will eventually form a stable population of cells known as a cell line. However, during this process, there will be certain characteristics of the original tissue or the organisms that will be lost or altered, and hence leading to changes in that composition or the behavior of the cell lines as compared to the cells when they are in the original tissue. So this is what one of the things that actually happens in vitro culture is that there is a loss of these heterotypical cell-to-cell interactions. Okay, so what actually happened? How does this actually happen? First, you're talking about selective growth, right? If you have an in vitro cell culture condition, certain set of conditions often favor the growth and survival of certain types of cells over others. As a result, when you establish a cell line, only the cells that can adapt and proliferate under that specific culture conditions that you have provide will survive and dominate the population. This selective growth can then lead to that loss of less robust cell types that are originally present in the original tissue. Okay, this whole process will actually lead to that absence of cellular diversity. Besides selective growth, you can also talk about altered microenvironment. In vivo, the cells are actually surrounded by a very complex microenvironment. So it includes extracellular matrix components, neighboring cells, and a plethora of signaling molecules. And these factors play very essential roles in regulating the cell behavior and function through the various cell-to-cell interactions. However, in vitro cell culture systems, you often lack this complexity that you can find in the in vivo microenvironment. So hence, you will lose certain heterotypic cell-to-cell interactions that actually depend a lot on very specific microenvironmental cues. Lastly is the genetic and epigenetic changes. So during the process of establishing a cell line, generally cells will undergo naturally. Okay, naturally, they will undergo genetic or epigenetic changes due to factors such as a prolonged culture or your suboptimal culture conditions or just inherent genetic instability. These changes can then affect the expression of genes involving cell-to-cell interaction, hence leading to that alteration in the cellular behavior and communication. Another thing to note is that in vitro cell culture also is lacking the original blood circulation that you find in the 3D in vivo system. Okay, so that's one thing extra to take note of. And also at the same time, they lack the homeostatic regulations because you have the endocrine system, nervous system within the in vivo setup, but you cannot find that in vitro. So they don't have that the homeostatic regulation that naturally happens in a 3D system. Section 3.12, Advantages and Disadvantages of in vitro cell culture. There are four major advantages using the in vitro cell culture system. The first two is the control of physical chemical environment and physiological conditions. In vitro cell culture actually provides researchers with a very controlled environment, okay, where various experimental conditions can actually be manipulated and monitored by you, such as temperature, pH, nutrient availability, substrate materials, and also the presence of different signaling molecules that you can add in the media to control or to study specific aspects of cellular behavior or their responses to different kinds of external stimuli. So basically, this system allows researchers to actually isolate and analyze individual variables in order for them to actually understand the different complex biological processes. Okay, so you have standardized protocols for cell culture techniques that allow researchers to always handle the cells and maintain the cells in a very consistent manner, hence minimizing variability and also enhancing the reliability of all the experimental outcomes. And this reproducibility is very important, okay, because it is really needed to validate scientific findings and also to advance research in many different fields. The other 2 points, which is the reduction of cost and also reduced animal use, is equally important because in vitro cell culture, it offers an alternative to animal experimentation for you to actually study basic cellular processes, disease mechanisms, drug testing, or any toxicity screening. It is way cheaper, definitely way cheaper. And also, if you were to use cell-based models compared to animal studies, you can actually address also the ethical concerns associated with animal research and also to minimize the use of animals in scientific experiments. Okay, as I mentioned before, cell lines, they're definitely way cheaper models to use. And also when you are using these cell culture models, it does allow us to study using human cells and tissues instead of animals. So you can have cells that are derived in the cells that are derived from human tissues and human samples. So it does provide researchers a more relevant and also translatable data for human health and diseases. There are 5 disadvantages, I would say key disadvantages when you're working with in vitro cell culture models. First is that the media. Okay, in vitro cell culture requires the use of cell culture media. Why? Because these media actually provide the nutrients and the growth factors and all other essential components that is required for cell growth and survival. However, these cell culture media really need to be changed regularly in order for you to maintain an optimal conditions for cell growth. This process can actually be labor intensive and time consuming, particularly when you guys are working with large number of cell cultures and also when you're performing very long-term experiments. Additionally, if you were to do improper media change, you can actually disrupt the balance of the nutrients and the growth factors, and hence this will affect the cell viability and also your experimental outcomes. Second would be the strict aseptic working conditions that are required. Okay, because you know that maintaining sterility is very, very crucial in vitro cell culture. Reason? The reason being to prevent contamination by microorganisms such as bacteria, fungi, and mycoplasma, which can then actually compromise your experimental results and also jeopardize your cell culture integrity. So to achieve and maintain these strict aseptic conditions really requires your meticulous attention to detail. Okay, and you have to really adhere to sterile techniques throughout the whole aspects of cell culture. Any breach in any of these aseptic techniques would then lead to contamination and then of course, creating delays or setbacks in researchers' research. Thirdly, high expertise. Successful in vitro cell culture requires a very high level of technical expertise and proficiency in aseptic techniques, cell handling, and culture maintenance. So you do have to possess a very deep understanding of cell biology, culture protocols, and must be very good at all your troubleshooting strategies in order for you to effectively grow and manipulate your cells in vitro. So you also need to know how to optimize your culture conditions for specific cell types. Okay, this may require a lot of very extensive experimentation and also optimization. So the skills are really advanced. You need really high expertise skills. Okay. So actually, we are hoping that when you take up this subject, cell technology, it is one of the entry to your training to have that skill sets. Okay. Fourth is the frequent subculturing. Okay, in vitro cell cultures, they have a very limited lifespan and they can undergo senescence or cell death over time if they are faced with factors such as overgrowth, nutrition depletion, or there's a lot of accumulation of waste products in your cultured media. So in order for you to maintain healthy and actively proliferating cells populations, your cells need to be subcultured regularly by detaching them from the current culture flask, and then you use some enzymatic or mechanical methods, and then you have to replate them again into fresh culture vessels with fresh media. Okay, so this frequent subculturing sometimes can be disruptive to cell physiology and could be one of the parameters that actually induce changes in cellular behavior, morphology, and gene expression. So sometimes it does has the potential of affecting your experimental consistency and reproducibility. Okay, so then with all these things, right? All these procedures that is required in vitro cell culture system, it actually may lead to certain differences in the phenotype and genotype of the cells that you are growing. Okay, so despite all the efforts that we try to mimic these physiological conditions that are found in vivo, cells grown in vitro will actually exhibit certain degree of differences in their phenotype and genotype. Okay, and these differences can arise from many different factors, such as their internal alterations in their cell signaling pathways, their gene expression patterns, their epigenetic modification, and how they actually interact with the extracellular matrix. So all these differences will actually happen because of all these alterations. And as a result, your in vitro cell cultures may not fully recapitulate that complexity and functionality of your cells within intact tissues or organs. Okay, so basically researchers must carefully consider all these limitations, okay, and try to leverage on other complementary approaches, okay, so they can use all these other complementary approaches to validate their findings that they find in the lab using the in vitro culture system. Section 3.13, Major Types of In Vitro Cell Culture. Primary cells, or also known as primary cell culture, refers to cells that you directly isolate from living organisms, such as from tissues or organs. And these cells are basically harvested directly from these biological sources without much manipulation or immortalization in cell culture. So they are all direct from the animal or humans. Okay, and primary cells, they retain many of the characteristics and properties of the cells within the original tissue or organism, including their physiological functions, morphology, and also gene expression profiles. Okay, so once you derive these from intact or dissociated tissues, they are then cultured, but after a certain period of time, they need to be subcultured or passaged. And this is what happens when you do that first subculture or first passaging, the primary culture becomes cell line. Okay, this is what they are known as cell line. Okay, and cell line, sometimes they may be called secondary culture. Okay, and a cell line is generally a single type of cell. Over time, as you keep on passaging them, you will get a single type of cells. So this diagram actually shows you the origin and progression of cell lines with the population doubling level. So you can see at the top, the tissue, you harvest some cells or some tissues from the organ fragments, you grow them, and that is your primary culture. Okay, you proliferate, let it proliferate, and then your first subculture, when you subculture them, they are the cell line. And if you just keep growing this cell line without any immortalization or any genetic manipulation, over time they will reach senescence. But if you want to create a continuous cell line, you have to allow them to go through certain manipulation processes before they become a continuous cell line and even transformed cell line. Primary cells have a finite lifespan in vitro, and they will actually stop dividing and in the end undergo cell death. And as a result, primary cells are often used for your short-term experiments or studies that require cells with characteristics that are closely resembling those of the in vivo tissues. So once these cells become confluent or they reach confluence, right? That means there's a lot of cells on the flask that you're growing. A monolayer or a cell suspension may be dispersed and they'll be reseeded and subcultured into fresh vessels. So the procedure that you'll be going through with these confluent cells is known as passaging or subculturing. As mentioned previously, after your first subculture of the primary culture, it will then become a cell line. And there's two types of cell lines, the finite cell line and the continuous cell line. A finite cell line is also known as a non-immortalized cell line, and it refers to a population of cells that have been cultured in vitro for a limited number of passages before it reaches senescence or it undergoes replicative exhaustion. Okay, so finite cell lines have a very finite lifespan in culture because you can only use it for about a certain period of time of your research. Okay, once they undergo death or senescence, you basically have to thaw out another cell line and continue with experiments. So this is the finite cell line. Then you have the continuous cell line, and continuous cell line, they refer to a population of cells that have been cultured in vitro, but it has the ability to proliferate indefinitely under specific cell culture conditions. Okay, unlike the finite cell lines, which have a very limited lifespan, continuous cell lines are immortalized and can be maintained in culture indefinitely through many generations. Okay, and there are various methods for immortalization. It includes viral transformation, chemical treatment, genetic manipulation, and these immortalization techniques often involve introducing genetic alterations or mutations that do what? Can guess? If you can't, basically these alterations or mutations can override the normal cellular mechanisms that regulate senescence or cell cycle arrest. Okay, so generally cells will go through senescence, right? But this mutation will allow the cells to override this mechanism. Okay, so this will allow the cells to continue proliferating beyond their normal lifespan. Okay, some advantages of continuous cell lines is that you have unlimited proliferation of cells. So basically almost limitless availability, right? Because they are able to proliferate indefinitely under the appropriate culture conditions, allowing for generation of large quantities of cells for experiments, screening assays, and also for production purposes. Next would be consistency and homogeneity. Continuous cell lines can maintain a stable and homogeneous population of cells with very consistent characteristics over time, making them very suitable for reproducible experiments and also large-scale studies. Third would be scalability. Continuous cell lines can be easily scaled up to accommodate the needs of experiments that require high throughput assays, drug screening, or bioproduction applications, okay? And they really provide a very reliable and very cost-effective platform for researchers to use. The disadvantage is that you will have genetic alterations and mutations, which then affect cellular behavior, their growth characteristics, and then hence leading to these cells retaining very little of the original in vivo characteristics. So where are we going to get our cell lines from? So basically we have cell banks, okay, cell banks carrying a large selection of very well characterized cell lines. Both American Type Culture Collection, ATCC, and the European Collection of Authenticated Cell Cultures, ECACC, are well-known and reputable repositories of cell lines. These organizations have extensive collections of very well characterized cell lines from not only humans, but you can get from many different species, different tissues, and also different sources, making them really a valuable cell banks for researchers to get their cells from for their research. And researchers can access their extensive catalogs online and also order the cell lines directly from them. Okay. Cell morphology refers to the physical structure, shape, and the appearance of cells when you observe them under microscope. Okay, it encompasses various features of cells such as the size, the shape, their organelle distribution, and also their surface characteristics. And studying cell morphology is very essential for your understanding of cell biology and physiology. Okay, because different cell types will exhibit different morphological characteristics that will reflect their functions and their states. So you can see here, there are a variety of cells. Some examples I'm showing here. You can see how different they look when they are actually grown on the monolayer in vitro culture system. So you can see some of them can be grown in suspension, so they're just floating in the media, or some of them they actually grow in monolayer attached to the culture flask. So these are some of the examples of how fibroblasts, neuronal cells, endothelial cells, and also our white blood cells look like under the microscope. You can use these questions after your lecture review to test your knowledge and understanding from the lecture. With this, I conclude section 3.1 of topic 3, cell technology. Hello students, this is the continuation of topic 3 lecture on cell culture and medium. In the next few series of lectures, I will be focusing on factors that promote cell growth. At the end of the series of lectures on this topic, you should be able to have a deeper understanding on cell cycle and in vitro cell growth, factors that influence cell growth in relation to cell culture medium and its constituents, and also the use of serum in cell culture. Section 3.21, Introduction to Cell Cycle and In Vitro Cell Growth Behavior. The cell cycle is made up of 4 distinct phases: G1, S, G2, and M. It is at the G1 phase where the checkpoint lies, where the cells decide to either progress towards DNA synthesis and another round of cell division cycle, or to exit the cell cycle reversibly, which is considered the G0 phase, or to irreversibly committing to differentiation. The proliferative activity of normal cells typically follows the sigmoidal pattern, characterized by 4 distinct phases: the lag phase, the log phase, the plateau phase, and the decline phase. The lag phase is the period of adaptation to the culture environment. The log phase is the period of exponential proliferation of cells. The plateau phase, which is the period when there is no net increase in the cell number. And finally, the decline phase is the period where cell death exceeds cell division. The lag phase is usually the initial stage, around 1 or 2 days following seeding and subculture. And there are some key processes that happens during lag phase. Number 1 is adaptation. The cells will replace the elements of its cell surface, such as their glycoproteins and also the extracellular matrix that were initially lost during trypsinization. Secondly is the attachment to the substrate. So this is a prerequisite for cell proliferation, particularly for the anchorage-dependent cells. They need to sit on the substrate and slowly get acquainted to it, and that is also part of the lag phase that's happening. Okay, and the third one is cell spreading. So once they feel comfortable, they will slowly start to spread. Fourth is the amount of DNA polymerase inside them will increase, followed by the synthesis of new DNA and structural proteins within the cells. And finally, there is no cell division during lag phase. So here is a schematic figure showing the top view and the side view of cells when you inoculate them into a cell culture flask, what happens after 1 to 2 hours where they start attaching, and then about 24 hours they start to spread out. So this is what happens during the lag phase in particular. Anchorage dependent, also termed as substrate dependent cells, they need to attach to a surface or a substrate in order to survive. Okay, this is a common one which is found in most normal cells because they need that attachment in order for them to start proliferating. The alternative to this is the anchorage independent cells because they are able to proliferate in fluid suspension or also in semi-solid medium such as agar or methyl cellulose. These are generally cells that prefer to grow in suspension, such as hematopoietic cells in your blood. Log phase is the period where there is an exponential increase in cell number. Okay, so the cells are now feeling comfortable in their cell culture environment, so they start to proliferate. So some of the general characteristics of cells during this phase is that its growth fraction is really high. It's about 90 to 100%. And culture is in its most reproducible form, meaning the cell numbers is just gradually increasing nicely. Okay. And thirdly, this is the optimal time for sampling, since the cell population is at its most uniform and the viability of the cell is really high. So if you want to do sampling to know the number of cells in your flask at certain time period, the log phase is the ideal time to do your sampling and cell counting. At the plateau phase, the culture becomes very confluent. Okay, usually this is at the end of the log phase, and all the available growth surface is already occupied. So some of you may have seen this in your practical classes. Okay, the characteristic features of this phase is that the growth fraction is around less than 10%, generally about 1 to 10%. There's less motility, cells can't really move. Cells occupies less surface area of the substrate and hence presents less of their own surface to the media that is in the culture flask. There is increase in synthesis of certain specialized proteins compared to structural proteins for them to stabilize in the cell culture flask. Hence, due to these changes in the expression or the synthesis of these proteins, this has resulted to the change in the constituency and also the charges on the cell surface. And lastly, cell number is generally in a steady state, meaning the number of cells that are dividing are actually balanced by the number of cells that is dying. One important metric to describe monolayer cell culture is confluence. So you all need to really be familiarized with the word confluence or cell confluency. It is actually telling you the percentage of the culture vessel surface area that actually get covered by the layer of cells that you are growing when you are observing them under the microscope. So for example, if you were to look under the microscope and you see half of the surface area of your culture flask is covered by cells, then you would say that the cell population is considered to be at 50% confluence. Okay, so if you were to look at the diagram on your left, okay, this is a schematic showing 4 fields of view under a microscope of cells. Okay, of cells showing 20%, 50%, 80%, and 100% confluence. So if you were to come and tell me, my flask has 100% cell confluency, that means your whole entire culture flask has full layer, a monolayer of cell culture that is growing. There's no more space. Okay, so at 100% confluence, if you were to look at the diagram on your right, the cell actually occupies less of the surface area of the substrate. Everything gets bunched together. And then it presents less of its own surface area to the medium. So it's really crowded. Okay, so that is what it means by 100% confluence, really totally all covered. When you are doing your subculturing, generally, you will have to subculture your cells when they are at 75 to 80% cell confluency. The level of cell confluency is really important because the cells are going to change their growth with changing density. So it's really critical. So generally, low density cells at about 10 to 20% confluency, usually they will grow slower than cells that are at 15% confluent. So if the plate is or your flask is completely grown by cells, right, it's almost reaching to 100% confluency, then they tend to grow slower again. So actually the level of confluency of cells in your flask play a kind of like play quite a big role in influencing the cell's genetic program and also how they actually behave in experiments. So you have to be really cautious or conscious, I would say, about the confluency of the cells that are growing, particularly when you are actually doing an experiment. It does influence how your cells are going to behave, and hence will result to your experimental results to be different. Okay, if you are using different kinds of densities, you're not consistent with your density, your experimental results will be not consistent either. Last but not least is the decline phase, where it is the period of cell senescence. One characteristic feature is that the cell numbers start to decline due to uncompensated cell death. Too many cells dying, but not many cells are actually proliferating. So why do cells die? So the cells at this point, where they actually reach this decline phase, they begin to die off for many reasons. So some of the reasons is due to lack of nutrients, right? The nutrients in the cell culture media have been used up. So lack of nutrients. Altered pH conditions. Why does this happen? Because there is accumulation of toxic waste products over time that is being secreted by the cells. So this environment, or I would say the cell culture microenvironment, is not really nice for the cells. So the cell starts to die. Okay, and hence it's critical in cell culture, you need to know how to do viability cell count, because you need to know. Sometimes you can't see. You see it under the microscope, you can't really tell whether the cells are healthy or not, right? You can't really have a number to tell you, okay, how many cells in my flask right now is viable. So you do need to learn how to do viability cell count. You can use these questions to test your knowledge and understanding from the lecture that I've just given. With this, I conclude section 3.21 of topic 3, cell technology. Hello students, this is lecture 3.22 covering on medium and constituents. So what is a culture medium? A culture medium is the media that we use to culture our cells, and it is also called the complete medium. Why is it called a complete medium? Because it basically contains nearly everything that cells need to grow well. So a complete medium generally is made up of two components combined together. One is a basal medium, and the other one is known as the supplements. So what exactly is in a basal medium? The first chemically defined media, known as the Eagle's Minimum Essential Medium, EMEM, was first developed in 1955. So this was the first widely used basal medium in cell culture. You can find basically basal medium available either in liquid, in a 500 ml or 1 liter bottles, or even in a powdered form that you can reconstitute with deionized water and sterilize it before you actually use them in the culture. There are two commonly used cell culture media that are found in the market. One is called the minimum essential medium, and the other one is Tobacco's modified Eagle medium. They are very commonly used in research labs for a variety cell types. However, you have to ensure that there are actually key differences between the two, particularly in the nutrient composition. MEM is a very basic cell culture media composed of very minimum concentration of nutrients that are required for cell growth in culture. While DMEM, on the other hand, is more complex media with increased concentrations of amino acids and vitamins that support the growth and maintenance of certain cell types. Here, if you use DMEM, you'll find that cells will definitely grow faster and they also deplete the medium slower. While both media are really widely used, it is also very important for you to choose the appropriate medium based on the specific needs of the cells that you are culturing. There are 9 key constituents in a basal media. You have fatty acids and lipids, accessory factors, amino acids, proteins and peptides, energy sources like carbohydrates, buffer system, inorganic salts such as the balanced salt solution, phenol rate, and vitamins. So what are inorganic salts? They are balanced salt solution, and they are defined as a combination of various types of inorganic salts that form a solution. So why are they critical in a basal media? They play several roles. First, they maintain the physiological pH in the cell culture media. They maintain the osmotic pressure of the cells. They maintain the membrane potential by providing enough sodium, potassium, and calcium ions outside the cell membranes. They act as cofactors in enzyme reactions and also aids in the cell attachment. Adhesion molecules usually need magnesium or calcium. There are many applications of balanced salt solution. First, they form the basis of many basal and complete media out there. Second, you can use them to dilute concentrated amino acids or vitamins while you're making the complete media. You can also use it for short incubation of cells like 30 to 60 minutes. Since most balanced salt solution do not contain nutrients required by the cells, of course they cannot support long-term growth or maintenance of the cells. So you can do short incubation in your experiments. And lastly, you can use it for washing or dissecting of your tissue samples or your cell samples because they are definitely way cheaper than using a complete medium. A buffer system is key in a basal media. As you know, cells produce carbon dioxide and lactic acid from glucose metabolism. So when you have a buffer system in the media, commonly it is actually the carbon dioxide plus bicarbonate system. It can help to compensate the effects that is caused by the acidic waste products produced by the cells to maintain that optimal pH within your media at the range of 7.4. So this buffer system actually helps to slow down that change in the pH. For a stable pH, many cell culture media will use the bicarbonate buffer system, which is actually the main physiological buffer system in our blood. Typically, the media will contain sodium bicarbonate, which will then balance with the carbonic acid and the carbon dioxide in the air. Increased acidity in the medium is generally manifested by an increase in hydrogen ions. What happens is that the free bicarbonate ions will then react with these extra hydrogen ions to form carbonic acid, hence shifting the reaction to the left, which will then stabilize the pH of your culture media. Similarly, when there is a decrease in hydrogen ions, it will result in a shift to the right, hence also stabilizing the pH of your cell culture media. You can watch this YouTube video which will explain to you much clearer the carbonate-bicarbonate buffering system. The major source of energy for cultured cells is the carbohydrates. Okay, most frequently used is glucose. You do have other kinds of sugars that are sometimes added in your culture media, such as maltose, sucrose, fructose, galactose, and mannose. But generally, you will find in culture media is actually glucose. At the same time, in the culture media, you will have L-glutamine. It is also another source of energy which is required by some of the cells. Even though L-glutamine is an amino acid and generally it is supplied to support protein synthesis by the cells. Proteins and peptides are supplied as nutrients in very low amounts in basal media because most of the proteins that are found in media are actually supplied by the serum. Some of the common examples of such protein supplements include alpha-globulin, fibronectin, albumin, and transferrin. And some of the main roles that they play is that they are actually carriers of minerals, fatty acids, and hormones. And they are also important in promoting cell attachment and also growth. They help to prevent toxicity. And at the same time, proteins and peptides also help to increase the viscosity of the media and also the cells. Amino acids are also critical for your mammalian cells because most animal cells do have this requirement for essential amino acids. So these are amino acids that are not synthesized by the body. So the cells cannot synthesize these amino acids. So these amino acids are then added into the cell culture media. At the same time, there are some other non-essential amino acids that are sometimes added as well in order to compensate for sometimes certain cell types that are not really able to actually make them in good amounts that they require for themselves. So some non-essential amino acids are added inside a cell culture media. Accessory factors is a blanket term that we use to cover all other types of additives that are necessary for effective cell growth. This includes things like trace elements because they are actually needed in very, very low amounts, but they are still essential for cell growth. So some of these examples are like iron, zinc, copper, and selenium. And all of these trace elements have their particular specific roles that they play. So you can probably read up a little bit more on how these accessory factors play a role in maintaining cell growth. Next, you have vitamins, lipids, and also fatty acids. Vitamins, you will have several different types of vitamins in the B group that are necessary for cell growth and multiplication or proliferation. And all these different vitamins are precursors for cellular metabolic cofactors. So they are really important for cells function, cellular function. Lipids and fatty acids at the same time, they are all supplied as nutrients and they are actually very important components of serum-free media. Cells are very dependent on lipids and fatty acids for many different kinds of cellular roles, okay, and also happenings that are taking place within the cells. They do need lipids and fatty acids. So these are really key nutrients that you have to add in your cell culture media. Generally, cell culture media is also supplemented with an indicator dye called the phenol red. So this phenol red is added as a pH indicator of the medium and it accounts for the color of your cell culture media. This indicator is really useful because it is particularly sensitive to slight pH changes around the growth optimum for cells. So a yellow hue to the medium indicates that the pH is too low or acidic, while a purple hue will show that the pH is too high, alkaline, whereas an orangey red hue will indicate that the medium is of the correct physiological pH. So sometimes you get an overnight change in the color of your cell culture from red to then yellow. This generally indicates bacterial contamination. You can go back and read and take your time to understand the content of this lecture on your own. And then you can use these questions to test your knowledge and understanding from the lecture. With this, I conclude section 3.22 of topic 3, Cell Technology.